Welcome to the ninth explainer in the series of research methodology titled The Objectives of Science and Research. I am genuinely so eager to guide you through this explainer today because we are unpacking a truly fascinating topic. When we think about the life sciences, whether we're talking about molecular biology, genetics, human physiology, or sprawling ecology, we aren't just looking at some rigid list of universal, unbreakable physical laws. It doesn't work like that. Instead, we are looking at a highly complex, slightly messy, deeply historical, and multi-layered engine that literally drives the living world. Think of it kind of like trying to reverse engineer an incredibly intricate machine while the machine is still running. Today, we are going to pop the hood on that engine. We're going to dive deep into the specific objectives that actually guide scientists in their pursuit of reliable, useful, and ethically accountable knowledge. It is going to be a great ride, so let's get into it. Here is our roadmap for today's explainer. We'll start with observing and describing the living world. Then we'll move into explaining how and why things happen, followed by predicting future behaviors and integrating those ideas. Finally, we'll tackle the immense responsibility of ethical intervention, evaluating truth, and the overarching philosophical frameworks that tie the whole shebang together.
All right, let's jump right into Section 1, Describing the Living World. The Foundations of Biological Data.
So let's dive into this very first objective, which is description. Now, you might be thinking that describing something is just passive, right? Like you're just making a catalog or a list of what's out there. But in the life sciences, description is actually a highly active, highly intentional practice. Consider this brilliant insight from the philosopher of science, Sabina Leonelli. She points out that open databases, things like GenBank or the Protein Data Bank, are actually an objective in themselves. They exist to ensure interoperability and reusability across laboratories worldwide. What does interoperability actually mean in practice? It basically means that a lab in Berlin and a lab in Boston are using the exact same biological alphabet. They can seamlessly share massive amounts of data without any translation errors. When biology shifted from just describing what an organism looks like, its morphology, to mapping out its actual molecular data, our descriptions began to carry very explicit evolutionary assumptions and background commitments. It's not just a catalog, it's a foundation. But here's the catch. To make all that descriptive data we just talked about actually useful, we have to do something called operationalizing it. Operationalization means making traits measurable in strictly standardized ways. Imagine for a second you're trying to study how fast a specific type of plant grows. If one researcher measures the height in inches every single Tuesday, and another researcher measures the weight in grams once a month, that data is totally useless when you try to combine it, right? Operationalizing means creating a strict standard protocol so that biological phenomena can be compared, integrated, and built upon globally. It creates the reliable, communicable accounts of living systems that we absolutely need. Because once we accurately describe life, the natural next step is that we must begin to explain it. Which brings us right to Section 2, Mechanisms, Histories, and Explanations, the how and the why. Explanation in the life sciences requires a deep mechanistic understanding of how organized parts interact within very specific contexts. And this beautifully illustrates a dual lens perspective famously articulated by the evolutionary biologist Ernst Mayr. On one hand, we have proximate causes. These are the immediate mechanisms acting during an organism's lifetime. It's the literal how. But on the other hand, we have ultimate causes. These are the historical evolutionary reasons why those traits exist in the very first place. That's the why. Think of a car, right? The proximate cause of it moving down the street is the combustion engine firing up and physically turning the wheels. But the ultimate cause is that human beings designed it for transportation. Or, to put it back into biology, think of shivering. The proximate cause of you shivering is your muscles contracting rapidly. But the ultimate cause, that's an evolutionary adaptation developed over millennia to maintain body heat so you can survive in the cold. To truly explain a biological phenomenon, you absolutely need both lenses. Building on that exact idea, the biologist Nico Tinbergen systematized these causes into 4 fundamental questions that provide incredible depth and rigor to any biological explanation. If we want to fully understand an organism's trait, we really have to ask 4 things. First, what is the direct causation or mechanism? Second, how does it develop? This is also known as ontology, which is really just the journey of an organism developing from a single cell into a fully formed adult. Third, what is its evolutionary history or phylogeny? Think of this as its family tree stretching back over millions of years. And finally, what is its function or adaptation? Any rigorous biological explanation aims to answer these 4 specific questions to get the complete 360-degree picture. So let's ground this abstract philosophy with a real-world example, okay? Let's look at the sickle cell allele.
If we want to explain sickle cell traits, we have to look at both the proximate mechanism and the ultimate history. The proximate mechanism, the how, is hemoglobin polymerization. That is the actual, immediate molecular process happening right inside the red blood cells that causes them to take on that distinctive sickle shape. But why does this trait exist and persist over generations, especially when it can cause health issues? That is the ultimate history, the selective pressure from malaria. Individuals who carry the sickle cell trait actually have a higher resistance to malaria, which is exactly why the genetic trait persists in certain populations where malaria is super common. You see how that works? We literally need the immediate molecular how and the evolutionary why to fully explain what's going on.
Moving right along, let's hit Section 3, predicting and integrating life, forecasting and connecting the big picture.
Okay, so prediction in biology is incredibly challenging. It's certainly a lot messier than in physics, but it is absolutely central to things like public health, drug design, and conservation. Today, we rely on several really powerful predictive tools. We use epidemiological modeling to forecast how infectious diseases might spread through a population over time. We use statistical information criteria like AIC and BIC, which are essentially complex mathematical tests to find the most accurate biological models among competing theories. And today, machine learning is vastly enhancing our predictive capacity in genomics by spotting patterns that human eyes could literally never see. But, and this is a big but, we have to heed the warning of philosopher Alex Rosenberg. He points out that prediction divorced from underlying explanation risks becoming a dangerous black box. Our goal in science isn't just blind algorithmic guessing. It has to be reliable, conditional forecasting tied directly to empirical validation.
And that brings us perfectly to integration. A truly distinctive aim of the life sciences is integration across multiple levels of organization. Biologists don't just look at one thing. They look at levels spanning all the way from microscopic genes, to cells, to whole organisms, all the way up to sprawling global ecosystems. The scientist Dennis Noble argues for a concept called biological relativity. This basically means that no single level here has absolute causal primacy over the others. You can't just say, oh, genes control everything and completely ignore the ecosystem. Nor can you look only at the ecosystem and ignore the cellular physiology. They all matter. Fields like Eco-Evo-Devo, which is a mouthful but stands for Ecological Evolutionary Developmental Biology, specifically work to link these distinct levels. They study how an organism's environment actually shapes its development and its long-term evolutionary path, integrating all these complex constraints together.
So, why does all this integration matter to us? Let's anchor this concept with a really urgent, real-world application, the One Health approach. One Health highlights that you literally cannot separate human health from animal health and environmental systems. If we want to tackle massive global challenges like antimicrobial resistance or zoonotic spillovers, which is when viruses jump from animals to humans, something we are all too familiar with now, we have to integrate our knowledge. Connecting molecular findings discovered in a sterile, pristine lab to global ecological observations out in the rainforest prevents fragmented science. And ultimately, that kind of integration saves lives.
Let's shift gears and maybe slow down just a bit for Section 4, Intervention and Ethical Design, going from knowledge to action. Because with the immense power to predict and integrate comes the profound ability to actually control and design life itself. There is a very critical tension in modern science derived directly from our source material today. On one hand, we have astonishing scientific capability. Researchers like Drew Endy describe the incredible goals of synthetic biology, which involves actually engineering genetic circuits and designing organisms from scratch to do specific things like produce medicine or create sustainable biofuels. It's a staggering capability. But on the exact same token, we have governance responsibility. Scholar Sheila Josinoff heavily stresses that these kinds of interventions demand democratic and societal oversight. Why? Because they don't just shape nature, they fundamentally shape human societies. The sources make it crystal clear. We have to balance the pure excitement of what we can do with a careful, neutral evaluation of how it should be governed. Because the interventions we are talking about here are incredibly high stakes. We're looking at things like gene drives. To put it simply, a gene drive is a technique that forces a specific genetic trait to spread rapidly through an entire wild population. Imagine altering mosquitoes so they can literally no longer transmit malaria. Huge potential, right? But we are also looking at germline editing, which changes DNA in a way that is permanently passed down to all future generations. Even in conservation biology, things like species reintroduction require weighing massive ecological, cultural, and political factors. So the crucial point you should take away from this is interventions in the life sciences are never just scientific objectives. They are deeply moral and ethical objectives that link laboratory capabilities directly to global responsibility.
Which naturally leads us to ask, how do we know we can trust any of this? Section 5. Evaluating truth in science. Safeguarding reliability.
With all these ethically loaded discoveries happening, how can we possibly trust the vast, churning amounts of information being generated? Well, listen to this massive, frankly surprising assertion from researcher John Ioannidis. He famously stated, "Most published research findings are false." Now, that is a shocking statement to hear in biomedicine. What he is pointing out is that due to issues like incredibly small sample sizes and something called publication bias, which happens because publishers love to print flashy, exciting new discoveries rather than boring, negative results, a lot of early findings simply don't hold up to scrutiny over time. This creates dramatic tension in the scientific community, and it is exactly why rigorous evaluation is absolutely critical as a core scientific objective. Science seeks not only to know, but to know reliably.
So, how do we fix it? How do we safeguard that reliability? Well, we do it by following incredibly robust evaluation steps. Step 1 is model comparison using predictive accuracy, ensuring we are choosing the mathematical models that actually align with real-world observed data. Step 2 is the triangulation of evidence. This means if diverse, completely independent methods all point to the exact same conclusion, we can be far, far more confident in it. And step 3 is sensitivity analyses. This involves essentially stress testing the findings by tweaking the variables slightly to see if the results still hold up, rigorously testing how robust they are under different conditions. Through these replication initiatives and open science reforms, we ensure that biological knowledge, while contingent and always evolving, remains trustworthy and, most importantly, corrigible. We can correct it when we're wrong.
Let's move into our final section, theories, paradigms, and conclusion, the overarching framework of discovery.
We've built this up step by step from basic description all the way to ethical intervention. Now let's look at the overarching philosophical theories that unify all these objectives. The life sciences are heavily theory-rich, and they are fueled by a truly fascinating philosophical dialectic. On one side, we have Karl Popper's concept of falsifiability. Popper insisted that for a theory to be scientific, it must be testable, meaning there must be a clear way to prove it wrong. But on the other side, we have Thomas Kuhn's idea of paradigms. Kuhn countered that science actually usually operates within accepted frameworks, what he called normal science, right up until revolutionary anomalies occur that the established frameworks simply cannot explain anymore. And you know what? The life sciences operate right in the middle of this exact tension.
Let's look at a perfect mini-narrative to illustrate how this plays out, the revision of the central dogma of molecular biology. For a long time, the central dogma, which basically says DNA makes RNA makes protein, was the paradigm guiding early normal science. It was the rule. But then falsifiable anomalies were discovered, things like reverse transcription, where RNA can actually rewrite DNA. Wow, right? But the paradigm didn't break, it expanded. Today, fields like epigenetics, which explores how your behaviors and environment can cause changes that affect the way your genes work, continue to shape modern thought. This brilliantly illustrates that biological theory is not about rigid, unbreakable laws. It is about dynamic unification. It accommodates anomalies and guides our future inquiry.
From describing foundational data to explaining complex mechanisms, predicting outcomes, and rigorously evaluating truth, the objectives of the life sciences generate not only profound knowledge, but incredible world-altering capabilities. So, I want to leave you with this final provocative thought. As our power to alter the very fabric of life grows, how will we govern the profound responsibility of our discoveries? It is a heavy question, but it's one we all must play a part in answering. Thank you so much for joining me for this explainer. If you found this breakdown helpful and you want to keep exploring the deep engine of science with us, please make sure to like, comment, share, and subscribe to the channel so we can keep diving into these fascinating topics together. See you next time.